Hi everyone, welcome to our weekly reading group happening every Monday on Zoom. Can just join us, hop on there and ask questions, discuss with the author, and all the information is down in the description. Yeah, and today we have Bowen Jing, a lab mate of mine here at MIT, and he'll be talking about AlphaFlow, where he generates ensembles of protein structures. So let's go.
Again, good morning, everyone, and thanks for the invitation and the interest. Today, I'm very excited to talk about our work on combining AlphaFold with flow matching, a recent generative modeling paradigm, which I assume many are probably quite familiar with, for generating protein ensembles. Now, as we know, AlphaFold and related methods have made revolutionary progress on the problem of protein structure prediction when framed as that of obtaining the best single 3D structure for a given protein sequence. But of course, proteins are not static. They don't have a single structure. They adopt an ensemble of structures with multiple states or potentially disorder that are involved in the functions of these proteins. And so there's emerged a new frontier in protein structure prediction, that of obtaining multiple structures from this ensemble using a computational method. Now, the existing approaches for doing so, or some of the existing approaches for doing so, have relied on the fact that AlphaFold is not a sequence to structure method, but rather an MSA to structure method. And so by messing around with the MSA, you can get AlphaFold to produce different outputs, which in certain cases can correspond to different conformations of the protein that are of relevance. And this modification can be done by subsampling the MSA, it can be done by mutating the MSA, or it can be done by clustering the MSA. And these three methods have all been shown to work quite well in many cases, actually. And these are definitely worth a closer look if you haven't read this body of literature before. But with that being said, there are some limitations to these methods that suggest they are not the end of the story. So for example, because of runtime considerations, it's recently become popular to have protein language model-based methods such as ESMFold or OmegaFold, in which case there is no MSA for you to subsample, mutate, or cluster. So how do you get different structures from a method like ESMFold? And additionally, and perhaps more excitingly, what if we want to model different conformational ensembles than the ones which appear in the PDB? So for example, if we have experimental data for NMR or other kind of data on disordered proteins or regions, or we have molecular dynamics simulations, well, those structures oftentimes are qualitatively quite different from anything that would appear in the PDB. And so there's no way that you will be able to get AlphaFold to produce those structures for you. So while these MSA ablation methods have done a pretty good job with making do with what we have right now, which is AlphaFold, it seems that there should be eventually a more principled way of actually training a model like AlphaFold for the purpose of generating conformational ensembles. And that's what we will aim to tackle in this work. So this is an emerging line of work, that of developing generative models for these protein ensembles. And just to all be on the same page, the problem definition that we're going to mainly be referring to is that of taking a single protein sequence and outputting a distribution of protein structures. The reason that we call this protein ensembles is to kind of disambiguate with the related problem of just generative modeling of protein structures for design applications, which is also very exciting, but it's a different problem because in that case, you don't have a protein sequence as input. But also on that note, I would emphasize that when we say protein ensemble, we just really mean a distribution of protein structures and not necessarily, strictly speaking, the Boltzmann ensemble, which is just a vocabulary thing to clarify. So there have been some other works in this area. Again, it's a very nascent line of research, but very exciting. And what we present is our approach to solving this generative modeling of protein ensembles problem is that of taking AlphaFold and repurposing it under a generative modeling training objective. So different from these prior works, we don't try to develop our own architecture. We just take the best off-the-shelf architecture that we have for producing an output protein structure, namely AlphaFold, and we just ask, what is the simplest modification that we can make to this architecture to make it generative? Okay, so with that, I'll talk about the method in detail a little bit more. So AlphaFold as a generative model. The key insight or the key fact that makes this modification possible is the observation that diffusion models or diffusion-like models provide a very simple recipe for predictive models to be easily converted to generative models. And to make this a bit more concrete, let's consider this graphic of a diffusion process, which we have probably seen many times. And we note that where does the neural network go in this process, right? The neural network is something that takes in a noisy image, in the case of image diffusion, conditions with some text input, and then outputs or tries to output a clean image. So the key observation here is that a generative model, in a diffusion sense, is nothing more than a model that, a predictive model, a text-to-image predictive model in this case, which additionally accepts a noisy image as input, right? So the only difference here is that we accept a noisy image as input. Nothing about the output needs to change. So similarly, if we have a sequence-to-structure model, we can have a sequence-to-structure generative model by additionally accepting a noisy structure as input. And so if we look at AlphaFold, we say, well, that almost actually looks like what AlphaFold already is, right? Because with AlphaFold, we output a structure, which tries to be a clean structure. We have a sequence input, and we also already have a structure input, in fact. Right now, AlphaFold is trained to accept template inputs. So all we need to do in order to make AlphaFold a generative model is to fine-tune it or retrain it, but in our case, we do fine-tuning. Fine-tune it to accept not a template input, but rather a noisy sequence as input. And we continue to expect AlphaFold to produce a clean output structure. And with that, we immediately get a sequence-to-structure diffusion model. Similarly, with ESMFold, there's also no problem here, right? Because ESMFold right now is a sequence-to-structure prediction model that takes in only a single sequence. But if we additionally fine-tune ESMFold to accept a noisy input structure, then we obtain a sequence-to-structure diffusion model. So I've been talking about diffusion so far, but the framework that we're actually going to use for fine-tuning is flow matching. And this is a slight generalization of diffusion, which many are likely quite familiar with, but just to all be on the same page, I'll walk through it again. So the general idea here is that you want to learn a flow field that transports your prior distribution to your data distribution. And the way that this flow field is trained and parameterized is via the following. So during training, you sample random pairs from your prior and your data distribution, and then you interpolate to a random point between them via some interpolation path, for example, a linear path. You feed this noisy interpolated image into your denoising model, and you try to get it to predict the clean image, the image from the data. Now, in reality, there are many interpolation paths that will cross. Recording in progress. Okay. There are many interpolating paths that will pass through this intermediate noisy image. So the model will never be able to deterministically denoise it, but rather it will learn something like the expectation of the clean image conditioned on the noisy image. And as you go through training and you cover the entire space and all different interpolation times, you end up with this vector field. So at inference time, this denoising model parameterizes the direction in which you should proceed if you wish to iteratively denoise data, right? So at the very beginning, you sample something from the prior, you predict using the denoising model, the expectation of the denoised image. You take a small step towards that. You feed that new interpolated image back into your denoising model. You get your new prediction, and you take a small step towards that, and so on. Now, to apply this to protein structures, we initially proceed in a very similar manner. We say, okay, we have our data distribution, which is just our database of protein structures. And we also need a prior distribution that takes the place of Gaussian noise for images. And for this, we're going to use a harmonic prior distribution, which has been used in a number of previous works to create random 3D structures that nevertheless have a polymer-like look to them, in the sense that adjacent atoms are close to one another, even in these completely random structures. So what we can do is we can sample a random noisy structure. We can feed this into our denoising model, which in this case is just going to be AlphaFold or ESMFold. And that will give us a target structure, a denoise structure, which we can then interpolate towards. We feed that back in to the denoising model. We get a new prediction. We interpolate towards it, and so on. Now, the picture that I've presented so far is a bit oversimplified. And the reason is because we cannot actually directly do flow matching with the structural coordinates. So in the previous visualization, we kind of directly carried the analogy from images to protein structures, treating both as a Euclidean space, in which we do linear interpolation and have an ODE in Euclidean space. Unfortunately, we can't do that directly, so let's take a closer look at the nuances that arise. So the reason that we can't do flow matching directly with structural coordinates is because the architecture itself is not able to propagate orientations from the input noisy structure to the output predicted denoised structure. So this is because we're trying to use AlphaFold and align to the AlphaFold architecture as closely as possible, make the minimal modifications. So what that means is that the input structure, it's going to be treated something like a template, right? And it's going to be immediately converted to a SE3 invariant representation of the noisy structure, which is going to be a histogram. This is what's currently done in AlphaFold with a template stack. And so that's what we're going to continue to do here. And then this information is passed through the eoformer and through the structure module. And then we have a 3D protein structure that comes out. But the problem is the 3D output structure has a completely arbitrary orientation, and AlphaFold has absolutely no way of propagating the orientation information from the initial structure to the final structure. So if you were to train with something like a mean squared error loss, this will just simply not work. There's just simply no way for AlphaFold to learn to denoise the structure, but keep it in the right orientation. So what this means is that we're going to have to redefine the space that we consider our protein structures to live in and adopt all of the properties of flow matching, or rather the steps of flow matching, the ingredients of flow matching to that new space. And so what this space is going to be is just the quotient space of your 3D point clouds under SE3, right? So this just means that point clouds that are roto-translationally equivalent to each other are defined to be the same object. And this is very sensible for AlphaFold because this is the space of inputs which AlphaFold is able to discriminate. So first we need to ask ourselves, how do we interpolate objects in this space? And because we're defining a flow matching framework, we're kind of free to choose whatever seems most natural. And we just define that to interpolate two roto-translationally equivalent point clouds, we simply do an RMSD alignment in the ambient space, and then we do linear interpolation after the RMSD alignment. In order to learn this expectation of a denoise structure, we actually notice that when we say we're learning the expectation in vanilla flow matching, what we're actually doing is minimizing a mean squared error loss, and that corresponds to learning the expectation of x0 given xt. So instead of minimizing a mean squared error loss, we just need to choose some equivalence loss, which is invariant, right? So that's how we learn this notion of expectation. And what we choose is the frame aligned point error loss, partially because this is what AlphaFold has already been trained with, so we know it has very nice properties. AlphaFold learns very well with this loss, but also because the frame aligned point error actually has the properties that we need in order for the minimizer of this loss that we're learning to resemble something like an expectation of x0 given xt. So with these modifications, the flow matching framework now looks something like the following. So during training, we're going to sample a random structure from our harmonic prior, and we're going to sample a point from the data. Now, the prior and the data may not be correctly aligned relative to one another. So the first thing we're going to do is RMSD align the prior sample to the data. Then we do our linear interpolation. We feed our interpolated structure into AlphaFold or ESMFold, which is going to predict an output structure, but possibly in the incorrect orientation, right? But that's fine because we then compute a SE3 invariant frame aligned point error loss, and we try to minimize that. So that's training. During inference, this prediction once again parameterizes the direction of a vector field, but now this vector field is not over Euclidean space per se, but over the space of R3 and quotient SE3. So we sample a harmonic prior, sample a noisy data point from the harmonic prior. We feed it through AlphaFold or ESMFold to get this putative denoise structure, which is possibly in the wrong orientation. We then reorient it, and we interpolate towards that in the ambient space. We feed this back into ESMFold or ESMFold to get a new prediction, which is again incorrectly oriented. We reorient it with RMSD alignment, interpolate towards it, and so on. So by doing so, we get a generative model which very closely aligns with the flow matching framework, except just in this new space, which is adapted to be maximally compatible with existing AlphaFold and ESMFold architectures. We have this general framework for turning AlphaFold into a flow matching generative model. And so now we want to evaluate on some ensembles. So we're going to do two sets of evaluations, one on PDB ensembles and one on MD ensembles. And by PDB ensembles, what I just mean here is that, even though we don't necessarily have an explicit ensemble for every entry in the PDB, there are many proteins or closely related proteins which do end up appearing in multiple PDB IDs with slightly different conformations. And so what we're hoping is to be able to, given a protein sequence, give us a number of structures which could plausibly have appeared by themselves in the PDB, even though there's not like a single entry that says this is the comprehensive set of structures for this protein in the PDB. Okay, so to train, or rather to fine-tune AlphaFold and ESMFold to do this task, we're just going to use the PDB, right? Except now with our flow matching objective instead of the standard regression-based frame-aligned point error objective. So we're training on the PDB for a healthy number of training examples. This is about 10%, roughly, the total training cost of original AlphaFold. And we're going to get these flow matching based models out, which we're going to call AlphaFlow and ESMFlow. And in doing so, we're going to be using the same cutoffs, the same training cutoffs as the original AlphaFold and ESMFold, so that we can compare results in a sensible manner. So for these PDB conformational ensembles, we have to ask ourselves, how are we going to do the evaluation, since we don't necessarily have entries of pairs of protein sequence and conformational ensembles. So what we're going to do is use the uniprot ID of the protein as a proxy for saying whether these are the same protein. And for each uniprot ID, you can find all the entries in the PDB that have a long stretch of overlap with that uniprot ID. That is going to be what we're going to call the PDB ensemble. And that will generally have 2 to 30 chains. We're going to draw 50 chains or 50 structures from our method and the baseline method, which is going to be MSA subsampling. And we're going to compute the precision, recall, and diversity, which are defined as follows. So to define the precision, we look at each member of the predicted ensemble, and we look at how close it is to the nearest true structure and compute its LDDT. And averaged over all of the predicted structures, this is going to be the precision. The recall is the converse metric. For every PDB structure, we're going to look at the LDDT with the closest predicted structure. And the average of that over all the PDB structures is going to be the recall. And finally, we're going to look at the diversity, which is 1 minus the LDDT between members of the predicted ensemble. And the 1 minus here is just so that for all of these metrics, higher is better. The test set that we're going to use is a set of recent PDB ensembles for 100 uniprot IDs, which have no representative chains in the training set, right? So on these 100 uniprot IDs, we do the evaluation that we showed on the previous slide. And here we're showing that for the baseline method, which is just AlphaFold with MSA subsampling. And AlphaFold is, you can sort of think of AlphaFold as already a kind of MSA subsampling, because by default, AlphaFold uses a maximum of 1,024 sequences in its MSA, and then 2,048 for the extra MSA stack. So if you have a very deep MSA, it's already doing some subsampling. And as you decrease that threshold more and more, that's the method that we generally refer to as MSA subsampling. And so what we see here is that as you decrease the depth of the MSA, as expected, you get more diversity and you get less precision. Strangely enough, on the right, you also see that you get less recall. So this just means that the model is actually not able to meaningfully improve, or rather not the model. So right now we're talking about the baseline here, right? But MSA subsampling is not able to meaningfully improve on AlphaFold in getting more of the structures in the PDB ensemble, which is a little different from the results that have been shown previously for MSA subsampling, but I think it's just a feature of this particular test set that we're using here. The main result here is that compared to MSA subsampling, when you run our AlphaFlow inference procedure, you get a much better precision diversity trade-off, right? So you get more diverse samples without losing that level of accuracy that you expect from AlphaFold. I mean, you do lose it a little bit, but not as much as if you subsample the MSA. And on the recall side, we actually have very similar behavior to MSA subsampling, which is that on this particular set of ensembles, we don't generally see that we're able to increase the coverage of the PDB ground truth structures that we get, but not in a way that is meaningfully different from an MSA subsampling. With ESMFold, there is no MSA subsampling baseline for ESMFold, right? So it's just ESMFold itself, which I would note, unlike AlphaFold, actually has zero diversity, right? So you may have noticed that AlphaFold here on the y-axis on the left actually has non-zero diversity, but ESMFold has zero diversity. And with ESMFlow, we're able to boost the diversity by a significant amount and boost the recall by a little bit, with actually very little cost in terms of precision. All right, so this is a significant gain in terms of these three metrics relative to ESMFold.
How do you control diversity during sampling? So what are the different?
Yes. Okay, so why is there a line? Right, okay, great question. So I didn't have a chance to talk about this in the inference procedure, but we actually, we introduce a procedure that we call truncated flow matching inference, where what you do is the first step that you take, the first integration step or interpolation step that you take, as it were, takes you all the way to time step 0.5 instead of 0.1 as you usually do. And so what this essentially does is it reduces the diversity but increases the precision at inference time. And the details are in the paper. Okay, so one interesting point that I want to highlight about what AlphaFlow is doing relative to MSA subsampling and why it's able to get a better diversity precision trade-off can be illustrated by looking at these PCA plots for these selected uniprot IDs. So what I've done here is just, done PCA on the set of structures from MSA subsampling, AlphaFlow and PDB pulled all together. And we can look at the PDB structures, the AlphaFlow samples, and the level of MSA subsampling that most closely matches the aggregate level of diversity of AlphaFlow. And what we see is that when you ablate the MSA, it creates more diversity, but it also causes the predictions to, in aggregate, drift away from the ground truth structure. Right, and this can be understood in the sense that the MSA subsampling is creating diversity by actually ablating the input. So it's sensible to think that that probably decreases the average input, the average prediction quality, and we see that manifest in terms of this this drift. Whereas for AlphaFlow, it is creating a diverse set of outputs, but it's doing so with the full set of input information and input signal that normal AlphaFold has access to. So AlphaFlow internally has the same representation of where the mean structure is, right? But all that it's able to do in addition to AlphaFold is to reveal what the variance of that distribution is by producing this ensemble of structures without incurring the drift that you would obtain by uploading the input. So with that, I wanted to show a couple of the conformational changes that we are able to resolve. So I would note that based on the recall metrics that we computed, neither AlphaFlow nor MSA subsampling are in general able to have this nice coverage of the multiple states of a certain uniprot ID in the PDB. But there are cases where we are able to do so. So here what I'm showing is in the black triangles and in the set of structures on the left, we have PDB structures for this particular uniprot ID. And on the right, we have the AlphaFlow structures. And I've shown 4 of them, but I've also circled the 2 that show the largest conformational change. So in this particular protein, you see that in the ground truth structures, which are labeled 1 and 2, in these 2 triangles here, we have this this little motif here. It kind of changes its orientation relative to the rest of the protein. And in the AlphaFlow structures, we also see a orientation change, although it doesn't quite go as far, which also matches what you see in the PCA. In this protein, this is actually much more subtle. So maybe this is not the best illustration. But what's going on here is that these 2 helices are changing in their orientation relative to one another. But unfortunately, it's very hard to see because they're one's behind the other. Maybe I'll not spend too much time trying to stare at this graphic. This one is a transporter, which of course, these proteins oftentimes undergo significant conformational changes. And here what we can see is the inner barrel of this transporter on the left is rotating a little bit between these 2 PDB structures. And we roughly see a similar rotation in the AlphaFlow samples. Now, eventually, hopefully, we'll be able to show these changes from a perspective that makes these changes a little bit more obvious to see. But for now, I've just circled them so that hopefully it brings it out a bit. These figures are, of course, all in the paper as well. So if you had questions about these specific changes, you can look there. Okay, so that I would move on to the MD ensembles, but if there are questions about the PDB evaluations, I can also answer them now.
We don't have any in the chat, actually.
Okay, all right. Okay, so moving on to the MD ensembles, I would clarify that there are actually two related problem statements that we're hoping to address when we do evaluations on these MD ensembles. So the first problem statement is what we're going to call the no templates problem statement, where just as in the PDB evaluations, we have an input protein sequence, and we're trying to output a distribution of protein structures. But there's also a related problem statement where, in addition to a single an input protein sequence, we also have a single protein structure. And we're going to call this a template structure, even though it's not necessarily used or interpreted in the same way as a template in the AlphaFold sense. It is a template in the sense that you assume knowledge to a single static structure, and now what you would like to do is to diversify that structure or study some conformational changes of that structure. And the reason why I highlight that these are two different problem statements is because they have different baselines and they have different use cases. So in the first case, the no templates use case, this is very similar to the PDB ensemble case, right? We have a single protein sequence, and without any other information, we want to get its full conformational distribution, ideally Boltzmann distribution, or as close to the Boltzmann distribution as we can get, right? And the baselines right now are just MSA subsampling, which has been used to some effect, for example, in AlphaFold Rave, as well as AlphaFold itself. In the problem setting where you do have templates, well, if you already have a single static structure, you can just run MD on it, right? So the baseline here is actually not MSA subsampling, but MD itself. So in some sense, this is an easier problem setting, but it's harder in the sense that we already have a very good baseline that we use and trust for this purpose. Now, I would also note that in order to make the architecture compatible with the second problem statement, we do need to add an additional input stack, which is going to be very similar to the input stack that we already use for giving the noisy structure to AlphaFold. Okay, so with these two related problem statements setting the stage, we're going to be using the same data set to evaluate both. And this is the Atlas data set from actually very recently published in Nucleic Acids Research. This data set contains explicit solvent all-atom simulations of a pretty healthy number of proteins, right? 1,390 proteins, which, at least to my knowledge, makes it the first molecular dynamics data set that tries to cover a good span of the PDB, unlike previous data sets well used in the community, like the D.E. Shaw research proteins, which are it's a set of just a handful of proteins, so not at scale. But here we have simulations for 1,390 proteins for the PDB. These are not terribly long simulations, but not terribly short either. We have a total of 300 nanoseconds of simulation per protein at 300 Kelvin. And importantly, each protein is representing a different ECOG domain. So the idea here is that this data set allows you to train on a split and then try to generalize to proteins that not only have not been seen in training, but are actually structurally dissimilar from those seen in training. So this is going to be a very important feature of this data set. So with that, what we're going to do is we take these ensembles and we split them by PDB release date. And this is just so that, again, we adhere to the same set of training cutoffs that ESMFold and AlphaFold were originally training. So that's May 1, 2018, May 1, 2020. Most of the proteins in the Atlas data set correspond to PDB IDs that were released before May 1, 2018. So we have 1265 training proteins, 39 validation, and 82 test proteins. And these don't add up to 1390 because there were some proteins that were just too long, and so we excluded them from the experiments. And this is like over 1,000 residues. Okay, so with this, a question that, as I understand it, has appeared in discussion a couple of times is 300 nanoseconds long enough? What is actually happening in these simulations? What are we learning? Is it meaningful? And so with that, I do want to be very clear. there are many use cases where 300 nanoseconds is just simply not long enough. And we are not going to be able to assess whether we can reproduce properties that you would normally expect to see if you run, for example, a 10 microsecond simulation. So what we will assess is precisely what we have access to, is the extent to which AlphaFlow can reproduce equilibrium fluctuations around the PDB conformational state. But critically, what we are assessing is whether we can do so in a generalizable manner. We're going to look at equilibrium fluctuations of some proteins and then try to predict the equilibrium fluctuations of unseen proteins. What we will not be able to assess is these rare transitions or conformational changes to alternate states that you would normally observe or expect to observe if you ran a really long simulation. We also cannot assess anything related to rollouts of trajectories because our model is a generative model. It's there's no ordering of the output samples, right? So we generate ensembles, not dynamics. And so if you're looking for any kind of kinetic information and whether we can reproduce that, it's just a completely different problem statement. So I just wanted to clarify, certainly 300 nanoseconds is not as long as you would like. I mean, ideally, you would love to have a millisecond simulation for every protein, right? But this is just the data that we have access to now. And what we're going to try to do is see to what extent we can use AlphaFold and ideas from generative modeling to learn from the data that we have now to reproduce, to serve as a proxy for doing the same kind of simulation, namely 300 nanosecond simulations on unseen proteins. Okay, so the training regimen to do these evaluations is going to build off of the model that we already obtained from training on the PDB. So we're going to take AlphaFlow and we're going to continue fine-tuning it on this Atlas dataset for 37,000 training examples. And we obtain a model which is like AlphaFlow, but now has seen and is theoretically able to produce conformations that are seen in MD, but could not appear in the PDB because those are typically at cryogenic temperatures. And this is a version of the model that we're going to call AlphaFlow MD. I want to highlight again what exactly is the nature of the generalization that we're testing, because it's a little bit subtle since we have essentially 3 stages of training, right? Pre-training from AlphaFold, the first stage of fine-tuning with AlphaFlow, and then the MD fine-tuning on the MD data. So the test proteins or ensembles that we're going to be assessing our generative capabilities on correspond to PDB IDs that themselves have not been seen at any stage of training, right? Because they were released after May 1, 2020. Additionally, they represent a different ECOD domain from any ensemble seen during the Atlas training stage. It is possible that they do not represent a different ECOD domain from any protein seen in training at any point, right? Because, essentially, that would be quite, well, that's just not how the data set was constructed. Right, so I do want to be clear about what kind of generalization we're assessing here. Okay, so the whole workflow is getting a little complicated, so I want to kind of lay out exactly how many different models we're currently dealing with when it comes to the MD evaluations. So there's actually 8 different models, right? Because there's AlphaFold and ESMFold, there's the no templates problem setting, and then there's the with templates problem setting. And also, to evaluate how much we can speed up the inference process, we train a distilled version for every base version of the model that we produce. So we actually have 8 different models that we could use to assess the MD evaluations, and all of these evaluations are in the paper. But for sake of just everyone's sanity, I'm going to completely exclude the ESMFlow evaluations from the talk. And within the AlphaFlow evaluations, I'm actually just mostly going to focus on the no templates problem setting with the base model. And later on, when we talk about comparing with short MD and whether or not we actually get a favorable runtime accuracy trade-off with MD, we're going to be looking at the with templates problem setting, because that's the setting in which running MD is actually an option. Okay, so we're going to walk through a number of classes of evaluations of increasing difficulty. But the first type of evaluation is simply to ask, can we model the flexibility of the protein in the sense that, flexible proteins have a lot of heterogeneity, rigid proteins have relatively less heterogeneity, and is the amount of flexibility well calibrated to what we see in the MD? And we can measure that by looking at the average pairwise RMSD between 2 members of the same structural ensemble. And we find that, at least at first, this very first evaluation at least passes the sanity check, that the AlphaFlow ensembles have pretty much exactly the same amount of aggregate diversity, as you would expect from MD. Now, with MSA subsampling, as expected, you kind of get this trade-off, right? The more that you subsample, the more diversity that you get. But the key point that we're looking for, which AlphaFlow is able to provide and MSA subsampling is not able to provide, is that the amount of diversity in an ensemble is predictive of the true amount of diversity in the corresponding MD ensemble. And we see that through this pairwise RMSD Pearson correlation here. Now, this is at an aggregate level, right? In the sense that we're just looking at the level of diversity in each ensemble. But we can drill a little bit closer and look at the RMSF, which is, of course, a very common metric when people look at protein flexibility. And we want to make sure that within a protein, for example, the more flexible regions are predicted to be more flexible and vice versa. And we see here an interesting phenomenon, which is that MSA subsampling, even though you get more diversity overall, it actually gives you pretty much no additional signal relative to baseline AlphaFold for which regions of the protein are more flexible. Whereas the AlphaFlow ensembles, they conform much more to the ground truth variations in RMSF as you move across the protein. So here is an example of that. We have here a protein and 10 samples from the predicted ensembles from AlphaFlow MSA subsampling, and then on the left-hand side, the reference MD. And we can look at the RMSF as a function of the residue index. And we see that the AlphaFlow ensembles have RMSFs that correlate very strongly with the ground truth RMSF. And not only do they correlate strongly, but are actually numerically about the same value as well. Whereas for MSA subsampling, the uncertainty from MSA subsampling comes mostly from the uncertainty the model has about the average position, right? Rather than trying to actually reproduce the ensemble. So for example, you see that this orange region is predicted to be the most flexible, whereas in the MD, that's just simply not the case. The green region is just as or even more flexible than the orange. So the second evaluation that we're going to look at is a generalization of RMSD. So we want to see for each atom in the ensemble, do we roughly capture the distribution of positions that that atom has over the course of the simulation? And so to compute that, we're going to define a quantity that we call the root mean Wasserstein distance, which is quite literally a generalization of RMSD in the sense that if you had ensembles which were point masses, this would just be RMSD. So we're going to fit three-dimensional Gaussians to the distribution of each atom in the AlphaFlow ensemble and then the MD ensemble, and then just look at the root mean Wasserstein distance with the mean taken over all the atoms in the structure. And by that metric, we see that the MSA subsampling is actually unable to give you a better idea of the distribution of the atomic position relative to baseline AlphaFold, whereas we are able to produce a lower RMWD. Now, the RMWD has a very nice property. It satisfies a kind of a Pythagorean theorem in that you can decompose it into a variance contribution and a mean contribution, where the mean contribution is how well do you predict the mean position of that atom. And the variance contribution is how well do you model the fluctuations of that atom around that mean position. And we see that what we are able to do is, we can improve upon AlphaFold in modeling the variance, certainly, since AlphaFold generally doesn't provide very much variance in a single atom's position. But we also improve upon the mean a little bit. Whereas MSA subsampling is just, it creates some diversity, but the diversity is just, it's not modeled correctly, and so we end up not even decreasing the variance contribution. And we also increase the mean contribution as a result of this drift phenomenon that we saw a little bit earlier. So another way that we can look at distributional accuracy is to ask, do we get the joint distribution of these atomic positions correctly? And one way that we can look at that is to do a PCA and extract the principal axis of motion, of collective motion of the protein structure, right? And what we can do is we look at this principal axis in the MD ensemble, and we look at this axis of motion in all of our predicted ensembles, and we compute their unsigned cosine similarity. And what I've shown here is a plot of for what fraction of the test ensembles does this cosine similarity surpass a certain threshold of success. So for example, if you say, I have recovered correctly the principal axis of motion, if my cosine similarity is above 0.5, I count that as a success, then we obtain twice the success rate for recovering the principal axis of motion relative to any level of MSA subsampling. which, as you can see, in general, is unable to improve upon even baseline AlphaFold in recovering this principal axis of motion. Just to give a little bit more context about this cosine similarity, this is the similarity between two vectors in 3-dimensional space. So 0.5 cosine similarity is already it's already quite large. Two random vectors in this high dimensional space would have cosine similarity pretty much always very close to 0. Okay, so the third set of evaluations, and perhaps the most interesting, is whether or not we can actually recover the phenomenon or the statistics of these fluctuations that we would expect to observe by running molecular dynamics. And the first set of such observables is going to be whether we can get transient and weak contacts of these proteins. So a transient contact is going to be defined as two residues which are not in contact in the crystal structure, but they do come into contact more than 10% of the time in the MD, and weak contact, vice versa. So here I've just shown two examples of such contacts, and we're going to see if AlphaFlow ensembles also give you the same information about which contacts have these properties. So the way that you can visualize what this evaluation means is by looking at the contact maps from MD and from AlphaFlow. And you can color these as follows. The regular contacts are just in gray, and then the red contacts are the transient contacts, and the blue contacts are the weak contacts. And you compute the Jacquard similarity, or IOU, of the sets of transient contacts between these two histograms, or contact matrices, and also the Jacquard similarity of the sets of weak contacts. And if the similarity is 1, that means that you have predicted exactly the correct set of transient contacts, and then if it's 0, the set that you predict and the true set of transient contacts has no overlap. So if we do this, what we see is we again are able to improve substantially over MSA subsampling in predicting the presence and the identity of these weak and transient contacts. And in particular, you see that MSA subsampling in the weak contacts case, it is able to offer some improvement over AlphaFold, but in transient contacts, just not at all, right? Which kind of makes sense, because if you have a weak contact and MSA subsampling is just adding noise, then yeah, it's probably going to tear the contact apart. But to get the transient contact right, you need to know, the flexibility needs to be going somewhere specific. And with MSA subsampling, oftentimes you're just not able to obtain that at all. So here I want to show some examples of these transient and weak contacts. I'm also showing here the percentage of the structures where the transient or weak contact is actually occurring. Because one question that you might have is, if I have my AlphaFlow ensemble and it predicts that this thing is a weak contact, and I see that it's in contact 20% of the time, does that actually mean it's 20% of the time? So in general, the answer is AlphaFlow is currently not very accurate at getting the occupancy rates of these states correctly. There are certain cases where it looks pretty close. Unfortunately, I cannot provide like a correlation coefficient, but I would just say that these two examples here are a bit better than average. But the point that I'm trying to make with these two examples is not necessarily the frequencies, but rather the kinds of motions that you do see with these transient weak contacts. So in these cases, we actually have two domains of the protein which are moving relative to one another. And you see that happening in the MD with some percentage of the time. And you're able to recapitulate that with the AlphaFlow samples, even though the crystal structure itself has a slightly different arrangement of these two domains. So the previous slide was focusing on these weak contacts. Here we have the transient contacts, which at least from the ones that I looked at are generally a little less interesting. Because these are usually just loop regions where there's a residue that's kind of close to the end of the loop, and the loop is sometimes over here, and then in the MD, when you heat things up to 300 Kelvin, the loop tends to disassociate and go somewhere else. So these two are not domain motions, but maybe still relevant if you care about, for example, some key residue on that loop. So the next evaluation we're going to look at is of a similar vein, but instead of looking at pairs of residues, we're going to look at individual residues and side chains and ask, can we identify which residues are going to be exposed to the solvent in the MD simulation, even though they were not exposed to the solvent in the crystal structure? So we're going to define the set of these exposed residues, or cryptically exposed residues, as those which have exposure to solvent more than 10% of the time in MD. In this particular example, this residue actually has exposure 86% of the time. And again, we compute the Jacquard similarity of the sets of residues from the MD and from AlphaFlow or from MSA subsampling. And we, again, get a smaller improvement this time relative to AlphaFold and MSA subsampling. But again, a pretty significant improvement relative to the amount of improvement that MSA subsampling is able to offer. These solid exposure analyses also allow you to do some even fancier probes and calculations. For example, from Greg Bowen's group, there's this concept of exposons, where you look at the mutual information content between the exposure status of residue A and residue B, and you can form this into a mutual information matrix, as shown here. And we can ask, how successful are we at recovering this mutual information matrix, which, as presented in this original article, is indicative of cooperative changes that are relevant for aspects of protein function. In this particular case, I've just shown an illustration of the AlphaFlow mutual information matrix and the MD mutual information matrix, so you can just kind of get a glance and decide for yourself if they're similar or not. Numerically, the Spearman correlation coefficient between the entries of these matrices is in general not particularly high. But I would also emphasize that this is quite a difficult task, and the MSA subsampling is really not able to give you this kind of information, whereas we are able to provide the beginnings of some signal towards this solvent exposure mutual information. Okay, so that is more or less the full set of evaluations that we carried out in the paper. I do want to briefly talk about what happens when you add templates in, right? Because we're going to be looking at some runtime evaluations. So with the templates, as expected, you generally get an increase in performance across the board. This improvement is particularly dramatic in modeling the level of global flexibility. So this Pearson correlation, once you add the templates, actually goes up to 0.9 or 0.94. I forget which one it is, in the sense that the aggregate level of flexibility that you predict for the protein is now almost perfectly correlated with the true amount of flexibility for that protein. And this is, you can sort of think of this as being because you now provide the model with the actual starting structure. So the model knows exactly which starting structure it's trying to predict 300 nanosecond fluctuations around. The distributional accuracy improves a little bit, both in terms of the atomic distribution as well as in PCA space, which is an evaluation that I didn't have a chance to talk about. And the MD observables, such as the weak contacts and the solid exposure, actually generally doesn't change very much. So here we observe the same performance. But the main point of running the evaluations with templates is to ask yourself the question, does this method offer any value to running MD itself in the setting where I already have a single protein structure? And this protein structure might be from the PDB, or maybe it's from AlphaFold, and I trust it as much as a structure from the PDB. If I can run short MD or I can run AlphaFlow, what would I do, right? And because we're using MD itself as our ground truth, it's kind of impossible to literally outperform MD, right? Since MD is the ground truth. So what we're going to do instead is we're going to assume fixed computational budgets, and we're going to say, if I can afford to run for this many GPU hours, do I get better results by running MD for that number of GPU hours or by running AlphaFlow for that number of GPU hours? And to answer that question, we essentially just plot the performance of each method as a function of the number of GPU hours. And so here, for these 6 metrics, we have the short MD in blue, we have baseline AlphaFlow in orange, oh, sorry, this is actually a typo. So the orange is AlphaFlow with templates, and then green is AlphaFlow with templates and then distilled. And so the distillation simply means that instead of 10 forward passes to do the flow matching, we only need to do 1 forward pass at inference time. And so it runs 10 times faster, but on some metrics, the performance decreases slightly. And so what we see here is that in general, for modest computational budgets on the order of 10 or fewer GPU hours, you actually do get better performance by running AlphaFlow instead of MD if your aim is to cover these aggregate statistics of the fluctuations around the starting structure. So, you get this faster convergence for many properties. But I do want to note, you don't get this faster convergence for all properties. So for example, in the solvent exposure mutual information matrix, for example, there's not really much improvement, as well as in the exposure Jacquard similarity. So it's not universally faster across the board, but for certain properties it is. Okay, so now I want to show some visualizations. And I want to preface with what exactly we're looking at here. So what we've done is we've looked at the 300 nanosecond simulation, and we, for each frame, we align it to the closest AlphaFlow samples, but we do so without replacement. And so this is in order to, also give a sense of whether or not AlphaFlow is getting the right proportions of the different things that happen in the MD. So these visualizations have dynamical information from the MD itself, just as a visual aid. The AlphaFlow samples themselves are not ordered. They have been post-ordered to align with the MD so that you can see something happen in the MD, and then you can look over and see if it's happened in the AlphaFlow sample. So this is the protein that's shown in the GitHub. The idea of these MD visualizations is to give the human eye a very fast way of iterating through a pretty large number of ensemble observables, which normally you would have to code up in a pretty tedious manner. So just looking at the ensemble, you can get a pretty good sense of whether or not the principal axis of motion is replicated. You can look at the different regions of the protein and how they're moving around, and you can check if they're moving the same way. You can look at the secondary structures, changing states from loop to helix, for example, and see if that's being replicated in the AlphaFlow ensemble. And in this particular protein, we get pretty good match between at least most of the properties that I've been able to visually inspect. We can look at another protein. So this protein actually has a quite larger range of motion because we have these two domains which separate for some non-trivial amount of time in the MD. And when we see that in AlphaFlow, we also do have the two domains pulling apart. It is clear that it's not pulling apart either as much or as much of the time as the AlphaFlow ensemble, as the MD ensemble. This is not to say that the furthest extremity of this motion is not replicated in AlphaFlow. I'm actually not showing every single frame here. So it's possible that somewhere in this animation, in one of the frames that's not shown, AlphaFlow does have a frame where the two domains are very far apart, but there's just not enough of them to cover this linear matching with the MD. And so we roughly see the flavor of the motion is correctly captured, but the extent is a little bit larger in the MD than in AlphaFlow. In this protein, we have a similar case going on. We have a fairly rigid domain in the bottom, and we see that in AlphaFlow, we also captured that rigidity, whereas at the top, we have a very disordered domain. Here, the domain is a little bit more disordered in the MD than in AlphaFlow, but again, the main motions are roughly captured, right? We have that this hinge-like structure is opening up, whereas this yellow loop on the side is kind of like an interesting latch like figure. And as someone who doesn't know protein biochemistry super well, I'll leave the interpretation of what this ligase is actually doing with this hinge motion up to you. So here is an example where the gross domain motion is actually captured pretty well. We have here a larger protein where this red domain has some small fraction where it actually spends quite a bit of time away from the parent domain. And we see that captured here in the AlphaFlow. It might be a little hard to see because it's only 1 out of the 30 frames where this happens. But if you catch it, you should be able to see that the range of motions is quite comparable in this case. Okay, so to wrap up, I want to clarify a couple of discussion points on what we are claiming AlphaFlow can do and what it cannot do, and in fact is just simply not designed or trained to do. So what we claim it can do is that it can produce ensembles whose fluctuations roughly emulate those of equilibrium MD at the 300 nanosecond timescale, because that is literally what we trained on and what we evaluated on. Hopefully, in the future, when we get better data sets, we can reproduce these evaluations for longer timescales. But for the moment, we're looking at 300 nanoseconds. We claim and show that we can obtain a better runtime accuracy trade-off than running short MD if your interest is in computing certain properties or statistics of these fluctuations. And for that purpose, for getting statistics of these fluctuations, AlphaFlow can substitute for MD with a more favorable trade-off. Now, these ensembles that you get out, you are also free to continue exploring them with MD, right? So the method can be used to propose these conformational states that you can then explore with other methods. What AlphaFlow cannot do, and we are not claiming by any means it can do, because it's just simply not designed to do this, is substitute for MD in applications that require explicit energies, such as free energy calculations. It cannot literally sample the Boltzmann distribution, mostly because the systems themselves are underspecified, right? We don't have solvent atoms. We don't have membrane atoms. So, there is no well-defined Boltzmann distribution that we are technically trying to sample. We cannot provide dynamical or kinetic information because we generate ensembles, not trajectories. And, at the moment, it's not clear how you would integrate this with, the whole menu of biased, non-equilibrium, or enhanced sampling algorithms that people like to use in molecular dynamics. So, to summarize, AlphaFlow is designed, and we show that it can accelerate one specific application of MD. It's certainly not, I mean, if there were any misinterpretation, it is certainly not the case that we were claiming that it can substitute for MD in general. But we do want to emphasize that for this one specific application, we are the first to be able to show that we can do so in a transferable manner across different proteins, and in fact across structurally dissimilar proteins, as given by this ECOD domain similarity split. So with that, that's the conclusion of my slides. Thank you very much for your attention and interest. Please feel free to check out the GitHub. Hopefully the instructions are quite clear to run, and I'm happy to take any questions.